All right. All right. Hello, everyone. We'll just get started in a couple of minutes.
Right, yeah, we're just giving people a couple minutes to trickle in. Okay, I think we're gonna give just about one more minute before Deepa gets us going.
All right, sounds good. All right, we'll get started. Hello, everyone. Welcome to today's session of the Leading Edge Symposium 2022, and thank you all for joining us. Today is the first day of Systems Neuroscience talks this year, and we're going to be hearing from 5 speakers on a range of topics that include neural mechanisms underlying different behaviors, as well as new methods for studying neural systems. Each talk is going to be 12 minutes long, followed by 3 minutes of questions. So for the audience, if you have any questions, please put them in the chat or raise your hand at any point, and Monique and I will either read your question or we'll call on you at the end of each talk. So now I'll pass it on to Monique to introduce our first speaker.
All right, thank you, Deepa, for that nice introduction, and welcome everybody. So first, today we're going to hear from Chuchu Zhang, and we're going to hear a little bit about the molecular and circuit mechanisms of nausea. She's coming to us from Harvard Medical School. So whenever you're ready, Chuchu, take it away.
Okay. Hi, my name is Chuchu Zhang, postdoc in Steve Lieble's lab. Nausea is a sensation of visceral malaise with or without vomiting, and I'm sure many of you have experienced nausea before. Why do we have nausea? Eating is essential for survival, but highly risky as internal organs are exposed to potential dangers such as toxins. One of the defense mechanisms to get rid of the ingested toxin is through tasting, olfaction, and vision, so that you can avoid taking in potentially bad food. But once food enters the gastrointestinal tract, one of the key defense mechanisms is through nausea and vomiting. While neuroscience in vision, olfaction, and taste represent some of the key discoveries in the field, the understanding of interoceptive system is largely behind. So in addition, nausea and vomiting can also be evoked by pregnancy, cancer chemotherapy, diabetes, and migraine radiation, while current anti-emetic drugs have only limited efficacy. So understanding mechanisms of nausea may help address an important clinical need. Sensory pathways leading to nausea have remained largely mysterious, with several routes to the brain possible. The vagus nerve is a key body-brain connection that monitors the digestive, cardiovascular, and respiratory system. With the gastrointestinal tract, vagosensory neurons detect organ distension, gut hormones, and potentially communicate with enteroendocrine cells and the entire nervous system. The airpostrema is a sensory organ within the brain. Neurons in the airpostrema are outside the blood-brain barrier and can detect peripheral circulation. Electrical stimulation of airpostrema evokes nausea and vomiting, and as a result, it is known as the chemoreceptor trigger zone for nausea for over 50 years. And then we have the vestibular system for motion sickness. None of these pathways are very well understood. I am most interested in the chemosensory pathways for nausea, and the most intriguing sensory organ for me is perhaps the airpostrema, and it's where I started. The airpostrema is located at the base of the fourth ventricle, next to the nucleus of solitary tract, which is where the vagus nerve terminating, and the vagomotor nuclei, which provide the majority of the parasympathetic inputs to the digestive system. As I mentioned before, one of the key features of airpostrema is that it has fenestrated blood vessels, and unlike the continuous blood vessels that form the blood-brain barrier in most of the brain region. And this can be demonstrated by intraperitoneally injecting fluoro gold into the animals. And you can see it's highly concentrated in the airpostrema, but not in the nearby regions. The airpostrema cells are rich in receptor expression and have been reported to detect several peripherally produced peptides. But in addition to this humoral route, the airpostrema also receive inputs from the vagus nerve, as you can see in the GFP signals here. So the airpostrema is anatomically advantaged to detect signals in the blood, as well as in the GI tract through the vagus nerve. However, how do the airpostrema contribute to nausea is not understood at a molecular level. I will tackle this question first by understanding the cell types and the receptors expressed in the airpostrema. I would then investigate how different cell types contribute to nausea and on the basis of the behaviors and cell signaling, as well as anatomy and circular modulation. To investigate the cellular diversity of the airpostrema, we use single nucleus RNA sequencing in collaboration with a grad student in the lab. Airpostrema was directly dissected using anatomical landmarks. Using unsupervised clustering of neurons, using neuronal markers, we identified 7 neuronal clusters with at least some markers expressed only in the airpostrema. And this includes 3 inhibitory types and 4 excitatory types. We immediately noticed that there are many gut hormone receptors expressed in the airpostrema. For example, the satiety hormone amylin receptor expressed in cluster 1, and the glucagon-like peptide 1 receptor expressed in all 3 other excitatory clusters. Agonists for GLP-1R treats diabetes, but are notorious for inducing nausea as a major side effect. One of the subsets within the GLP-1R population also expressed receptor for GI-15. GDF15 is a stress response cytokine, which has recently been discovered to be increased during cancer chemotherapy, bacterial infection, and pregnancy. Some research have also shown that GDF15 can induce can suppress food intake, and in some animals, even nausea and even vomiting. So we validated functional expression of these receptors and generated or created knocking mouse lines to access small or large clusters of the airpostrema neurons. One of the manifestations of visceral malaise is avoidance behavior associated with ingested toxins. Mice do not vomit, but can develop powerful aversion associated with human emetics and can also be blocked by antiemetics. Briefly, mice will be exposed to either cherry or grape flavored saccharine water and immediately followed by injection of nausea stimuli. When mice are presented with the conditioned flavor again, it will avoid this flavor if it has previously experienced a visceral malaise, like you would avoid a food that makes you throw up. We have tested a lot of nausea stimuli, and indeed, cancer chemotherapy cisplatin, GI irritant lithium chloride, bacterial LPS, and the diabetes drug GLP-1 agonist, X74, all promotes conditioned flavor avoidance. And in this preference ratio plot, 0.5 and lower indicates avoidance. So we use chemogenetics to activate small excitatory neural clusters in the airpostrema and carry out a conditioned flavor avoidance test. Chemogenetic activation of neurons marked by GLP-1R, GFRO, SLC6A2, but not CALCAR, promotes avoidance conditioned to the paired flavor. And just to remind you that the airpostrema exciting neurons are largely divided into the GLP-1R population and CALCAR population, and only GLP-1R and its subpopulations can promote avoidance. To test whether avoidance promoting neurons are required for poison responses, we used a genetic ablation approach to ablate the GLP-1R neurons in the airpostrema. After ablation, mice failed to display avoidance conditioned to X74, lithium chloride, LPS, but not to cisplatin. This suggests that several visceral malaise cues act through the airpostrema for aversion. We also ablated smaller subsets of airpostrema neurons and found that there are subtype-specific responses. For example, for the lithium chloride-induced aversion, the GFRO population is critical, but not SLC6A2 population. So these two aversion-promoting airpostrema neuron types may engage distinct signaling pathways under different nausea conditions. So far, I have identified cell types relevant for nausea-like functions through chemogenetic gain of function and loss of function studies. Not shown here, but I also investigated the cellular physiology and signaling pathways relevant for the nausea conditions. Next, we look at the circuit level engagement and modulations. To understand what downstream neural circuits are engaged, I mapped projections of genetically defined airpostrema neurons. Airpostrema excited neurons project to multiple brain nuclei for gastrointestinal controls, such as the NTS and DMV, and cardiovascular controls, which is the RVLM. In the midbrain, it also projects to the parabrachial nucleus, which play important roles in aversive learning. On the other hand, the inhibitory neurons project only locally within the airpostrema and the nearby NTS. This is intriguing because the functions of airpostrema inhibitory neurons are largely unknown, and perhaps they can play a role in the modulatory functions of the excitatory neurons and maybe even inhibits nausea. Interestingly, the inhibitory neurons also express gut hormone receptors, including the hunger hormone ghrelin receptor and the glucose-dependent insulinotropic peptide GIP receptor. It has recently been shown that GIP can suppress adverse nausea-associated behaviors in response to GLP-1R agonists, and also can suppress cancer chemotherapy-induced vomiting in ferrets. But it's not clear how GIP can have such anti-emetic effects. We wonder whether we can find mechanistic basis for the reported anti-emetic functions of GIP. Using GIPR-CRE and a GIPR-CRE and a reporter, I created a whole cell patch count recording from brainstem slices. Perfusing GIP rapidly depolarized GIPR neurons, which was independent of any synaptic blockers, indicating a direct activation of GIPR neurons. Next, we ask whether GIP evokes inhibitory postsynaptic currents in particular classes of the air postrema excited neurons. So we used whole cell patch count recording from brainstem slices, which also express reporter for all the subsets of the air postrema excited neurons. In contrary to GIP-induced depolarization of GIPR inhibitory neurons, GIP-induced hyperpolarization in a GFRO expressing air postrema neurons, and also the GLP-1R neurons, which also include the GFRO neurons, but not much so in the other excitatory subtypes. The hyperpolarization can be blocked by GABA-A receptor by cuculin, suggesting that this hyperpolarization is through GABA. And here are some of the quantifications. So together, these findings indicate that GIP can directly stimulate air postrema GIPR neurons, which in turn form local and selective inhibitory currents or connections with the air postrema GFRO neurons. So can we also observe behavioral inhibition? In mice, administered either saline or GIP on the conditioning day, they were exposed to either the GFRO agonist GI15 or lithium chloride 20 minutes later. While GI15 and lithium chloride provides a robust aversion in saline-injected animals, a prophylactic injection of GIP abolished the aversive response. To determine the site of action, we ablated the air postrema GIPR neurons with genetic ablation approaches. The DT ablation was highly specific, and after we ablated the air postrema GIPR neurons, this protective effect is largely lost. So GIP suppressed nausea-associated behaviors through the air postrema inhibitory neurons. So in summary, we established the first cell atlas of the air postrema and probed for nausea mechanisms at the molecular level. We identified two excitatory populations that can directly induce aversion and are required for different toxin-induced visceral malaise. We identified distinct we also identified a distinct excitatory and inhibitory circuits in the air postrema. And finally, peripherally derived GIP can activate the air postrema inhibitory neurons expressing GIPR, which in turn suppress the air postrema GFRO neurons. And this can also function at a behavior level to protect the GFRO-induced aversion. We speculate that the air postrema circuits that integrate the reward and punishment signals could guide future consumptory decisions based on need and toxin risks. Altogether, we have uncovered the basic organization of the air postrema nausea circuit and provided a framework towards understanding, predicting, and therapeutically controlling nausea. Some of the future directions in my independent career include understanding the role of vagal neurons in nausea. This work has been focused mostly on the humoral detection of cues in the air postrema, but as I mentioned earlier, it also receives vagal inputs. We want to understand what do these vagal neurons communicate with in the air postrema, and what do these vagal neurons detect in the GI tract, and how do these vagal neurons may modulate activity of the nausea-promoting air postrema neurons. Another direction that I'm interested to pursue is to identify the signaling cues for nausea. The molecular mechanisms for nausea are still largely unknown and represent a key knowledge gap. I'm interested to investigate the signaling cues and sensory pathways of nausea that occurs during poisoning by bacterial toxins, but also occurs maladaptively during cancer chemotherapy, radiation, and other conditions. And with that, I would like to thank my mentor, Steve Lieblis, people that have contributed to this work, including grad students, undergrad research assistants, and postdocs. And I would like to thank my collaborators and advisors and my funding source. And also I like to thank Leading Edge for the opportunity to talk here. And I welcome any questions.
Thank you so much, Chuchu, for a wonderful talk. That was fascinating. I'll just pose one question to you, and that's how much do you think conditioned cues might play a role in this nausea pathway? And do you think that this pathway might be important for conditioning for other types of behaviors? Let's say reward, for example, like conditioned taste preference rather than aversion.
So the associated learning circuits is in the parabrachial nucleus, which then projected a central amygdala. So I think airport stream plays a role in detecting and basically give the inputs to engage this aversive circuits and learning. So I guess your questions, and then we also show that you can also kind of by blocking the activation of exciter neurons with the airport stream inhibitor neurons, you sort of disengage or stop the engagement of the circuit. So I guess any cues that can activate airport stream exciter neurons may potentially engage the circuits, and cues that engage the inhibitor neurons may potentially like disengage. So I wonder if I answered your questions.
Yeah, you did. Sorry, it was more like a multi-part question. I should have made it more specific, but no, that's great. I think it would be interesting to sort of parse that out in the future.
Yeah.
But just to keep track of time, I think we do have to move forward. So I'm going to hand it over to Deepa to introduce our next speaker. Thank you again, Chuchu. Wonderful job.
All right. So thanks, Chuchu. Our next speaker is going to be Xiaotang Lu from Harvard. And the title of her talk is ADECO, a whole brain staining method for connectomic studies. And yeah, take it away, Xiaotang.
Hi, do you see my screen? Okay, so my name is Xiaotang. I'm a postdoc working with Professor Jeff Wickman at Harvard. And I'm very honored to have the opportunity today to present my recent work of whole brain staining method for connectomic imaging. So 100 years ago, Cahal utilized a new staining method called Golgi staining to discover that neurons are interconnected in a directional network. His beautiful hand drawings of microscopic images still provide us with excellent representations of the essential of neural circuits. The only problem with these images is that they didn't represent the actual complexity of neural circuits. In reality, each neuron in the brain is maintaining thousands of connections with thousands of other neurons. This is absent from his drawing. A recent leap in our understanding of neural circuits was benefited by the development of serial section electron microscopy, the tissue staining, and progress in machine learning. As shown in this video, this method and tools conjointly allow us to image a brain tissue at nanometer resolution that covers all the cellular and subcellular details. So afterwards, we are able to reconstruct any or all the synaptically connected neurons from this image, EM image stack. This nascent field of neuroscience is now known as connectomics. We aim to map the actual neural circuits that underlie underlying brain functions. With advances in automated EM mapping over the past decade, we're now able to reconstruct the connectome from increasingly larger brain tissues. For example, this is all the pyramidal neurons our lab reconstructed from 1 cubic millimeter of human cortex, collaborating with Google. It is truly amazing to see such images. But one of the problem is, if you look at one of the neuron reconstructed from data set, even we are able to reconstruct all the passing axons that making synapses with it. Most of the time, it's still difficult to know more details about its partners because they usually reside outside the data set and sometimes from distal region. For this reason, whole brain connectomes are required for us to understand how a brain works as a whole. This kind of mapping is now visible in small animals like worms and fruit flies, but only very small portions of mammalian brains are now visible for connectomic imaging. The largest connectome mapped from a mouse brain covers only 0.2% of its total volume. One of the problems is that staining brain tissues greater than 1 cubic millimeter is still technically challenging. So to get sufficient signal for EM imaging, we need to stain the tissue several rounds with heavy metals such as osmium tetroxide and uranyl acetate. But brain tissue is much more membranous than and heterogeneous than most other tissues. So intense and uniform staining I'm sorry, there's some technical issue. So intense and uniform staining is very difficult to achieve simultaneously in a large brain specimen. Worse still, a stained brain can only be assessed after finishing this 2-month-long procedure. If one step fails, we are unable to know until 2 months later when we look at the tissue under EM. So to solve this problem, I set up a reaction capsule inside an X-ray microscope so that we can monitor the staining process in real time by virtue of the high penetrating power of X-ray photons. This video shows the first osmication of a neonatal mouse brain at an accelerated pace. You can see clearly how the staining fronts migrating in the brain volume. With the help of time-lapse X-ray imaging, I developed a whole brain staining protocol that we called ODECO. The first thing I discovered is that osmication. So sorry. So the first thing I discovered is that osmication rate is not only size dependent, but also tissue dependent. A P0 brain takes only 4 hours for osmium to reach the center, but for an adult mouse brain, the first osmication took over 44 hours, even though the volume is only 6 times bigger. Besides knowing the optimal duration for staining, X-ray imaging can also be used to probe the reaction mechanism. For instance, potassium ferrocyanide is often added after first osmication to boost the contrast of membrane staining. It is thought that ferrocyanide can reduce osmium tetroxide to potassium osmate, which bias to stain membranes rather than cytoplasm. What I observed from X-ray is that the X-ray scattering decreased after applying ferrocyanide. It indicates to me that some the osmium has been removed from the tissue after the step. So ferrocyanide actually de-stained the brain. To figure out what portion of osmium was being removed, I made 3 test gel blocks to represent carbohydrates, lipids, and proteins. After first osmication, the lipid block and the protein block turned dark, but the carbohydrate block was unstained. It means that lipids and proteins can form the strong bonding with osmium, but the carbohydrate can't. After applying ferrocyanide, the first 2 blocks didn't change color, but the dark brown of the protein block faded away. This result suggests that ferrocyanide can dissociate osmium bound to protein. It therefore reduces estuary scattering, but helps reduce membrane-enhanced staining because it lowers background cytosolic staining. And because most of the binding sites has been saturated in the first round of the staining, to add more stains, we need to add a multidentate ligand that anchors to the existing metals, but provide additional sites for the second round of staining. TCH is widely used for small tissue staining, but we found it's impossible to use TCH for whole brain staining. The brain was filled with cracks after this step because TCH releases gases when reacting with osmium. Those gases cannot diffuse out of the brain and lead to cracks. Pragya Goel turned out to be a much better choices for whole brain staining. It doesn't bring about any noticeable artifact. After Pragya Goel, the brain is now ready to accept more osmium. What I observed is that second osmication is much slower than the first osmication, as shown in those videos. And like neonatal brains, the second osmication also takes 1.6 times longer for adult mouse brain, suggesting that existing osmium might form a diffusion barrier in this round of staining. I also investigated artifact formation during this long tissue processing. For example, it is known that dehydration is associated with volume shrinkage. Traditional dehydration involves a stepwise solution change from 25% to 50% to 75%, then 100%. It entails actually abrupt volume shrinkage at each change and might lead to tissue cracking. To enable a smoother dehydration and avoid a sudden disturbing to the tissue, I adopted a gradual dehydration strategy. However, when I dehydrate a P0 brain over 6 hours dehydration process and cracks forms. But when I extend it to 30 hours, cracks can be prevented. For an adult mouse brain, even 90 hours dehydration would cause cracking, but 120-hour dehydration wouldn't. A more interesting finding is that the tissue didn't contract until the non-polar solvent get to over 80%. So I guess the take-home message is that dehydration rate should also be adjusted to match the tissue volume. And with the help of X-ray imaging, we now have a whole brain staining protocol. So let's examine the EM quality of an Odeko-stained neonatal mouse brain. So this is the anterior part of the brain block. And the olfactory bulb looks beautifully stained, and we move to the center of the brain. The membrane is also look crisp. And if we move to the posterior of the brain block, the tissue is also well stained. So it proves that Odeko yields enhanced membrane contrast throughout the brain volume. And as all the steps can be scaled with time, Odeko can actually be successfully applied to both small tissues like 1 millimeter brain slabs and whole brains. And beyond the staining, I also investigate the volume changes during staining and rinsing, and also develop set of strategies to mitigate the formation of artifacts. Looking to the future, because connectomic is still developing, I want to tap into my past training in chemistry and material science to develop tools and methods for connectomic research. First of all, I will keep scaling up the staining and imaging pipeline to enable larger connectomes. and to address the need of research studying circuits that span several brain regions or covering both central and the peripheral nervous system. I plan to optimize the long-range tracing techniques to make them more compatible with sequent EM imaging. And as the complexity of the nervous system is also reflected in its molecular diversity, to better interpret brain functions from connectome, we need to integrate other information into connectomes, such as molecular identities of neurons. So how to realize that will be one of my main focuses for future research. Also hope to standardize the connectomic pipelines to make it an everyday tool for neuropathology. It will require developing protocols in collaboration with neurosurgeons in different brain regions and pathological states. The ultimate goal is to make connectome no longer an expensive tool restricted to a few labs in the world, but accessible for all the brain researchers. And we are moving in that direction. With that, I want to thank my mentor, Professor Jeff Wickman, and all my colleagues who helped with this project and my funding from Brain Initiative K99. Thanks.
Thanks, Xiaotang. We have time for a few questions, and we can open it up to the audience if anyone has any questions. I have a question for you, Xiaotang.
Sure.
So you've talked a lot about scalability in terms of volume, which is very important and a difficult problem in connectomics. I guess as kind of coming from a systems experimentalist perspective, a question I have is, there's also a lot of individual variability across brains. And so I was wondering about how scalable it is in terms of numbers of brains this procedure can be applied on simultaneously to be able to compare the connectome across individuals.
So actually, that's a really good question. So actually, we didn't find a lot of variabilities between different species and different brain samples. So that's why I aims to get the highest membrane contrast from all the samples I'm dealing with. So I mostly work on the mammalian brain samples, and all the brain samples can be well stained with my protocol. But in the future, I would love to look at brain samples from other species, such as fishes and some birds, or the marmoset, to know if there's any differences.
Thank you. Oh, okay. So we have a question in the chat from Anna. And she asks, what kind of approaches might you take to optimize cross-region connectomics?
That's a still experiment I'm planning. So one of my thinking is that because the current neural tracing uses reporters fluorescent proteins, and to get this long projectome across this whole brain volume. So some image methods we now take is to, for instance, clearing the homoscreen to look at to resolve this projection, or to cut several thin sections and align those image to trace the circuit. And those methods are not very compatible with EM because, for instance, the tissue clearing will remove all the membranes from the tissue, and membranes is a main component we're looking at with the EM. So what I'm thinking now is to first to optimize the reporter signals. So what I'm interested currently is to use alternative heavy metals to resolve this projection with the help of X-ray. but that is only one of the idea. I'm still like discussing with the neurobiologist to see if there's any other like interesting ideas I should try. Yeah.
All right, thank you. I think Monique, probably go to the next speaker.
Sure, thank you so much for that. All right, so next we're going to move on to Pragya Goel. She is coming to us also from Harvard Medical School, and she'll be talking a bit about signaling structure for dopamine coding in the vertebrate striatum. So take it away, Pragya.
All right, thank you. So can you guys see my screen? All right, so I'd first like to thank the Leading Edge committee for this opportunity to present my work. I think it's been an enriching experience so far to be part of this fantastic community of scientists. So I'd like to start by saying that a lot of beautiful work over decades has uncovered the signaling structures, meaning the precise release receptor organization in case of neurotransmitters such as glutamate and GABA. For dopamine, serotonin, and acetylcholine, this exact signaling structure is still unclear. And so as a postdoc in the Kaiser lab, this is what I wanted to figure out for the neurotransmitter dopamine. Okay, so we know that dopamine signaling controls diverse processes and behaviors such as movement and responses to reward, motivation, and learning. And correspondingly, defects in signaling result in pathologies such as Parkinson's, which is the most obvious one, and also addiction. And it's interesting that it mediates these behaviors on a range of time scales, from fast impulse responses on sub-second scales to store effects over seconds, hours, and days, suggesting that dopamine transmission has a fast component in addition to its known slow modulatory one. These behaviors arise from different dopamine pathways. We study the nitrostriatal pathway responsible for movement and motor control, where cell bodies in the substantia nigra in the midbrain send long axon projections to the dorsal striatum. So several studies over the last few years have led to this notion that dopamine typically operates by volume transmission, where diffusion of the transmitter from these axon swellings called varicosities into extracellular space activates widespread receptors over several microns. And this is in stark contrast to fast classical synapses, where release is specialized at these presynaptic active zones directly opposed to postsynaptic ionotropic receptors, where there's a tight alignment at the scale of nanometers in order to achieve rapid and precise signaling. So the very nature of transmission by diffusion for dopamine suggests that this kind of signaling structure with a tight spatial organization that underlies fast transmission might be dispensable. However, the timescales reported by functional and behavioral studies for dopamine are rapid. So how can this be reconciled with the fact that dopamine transmits by volume diffusion, which is inherently slow? So cellular and molecular work from our lab and others has recently shown that dopamine release in the striatum occurs with rapid kinetics at sparse release hotspots. And these hotspots are similar to those for fast exocytosis in that they use three key fast synaptic transmission molecules, namely RIM, MUNC13, and the calcium sensor synaptotagmin 1. All in all, demonstrating that the machinery for fast signaling is present in these dopamine axons, at least presynaptically. So this raises the question that if there is a sophisticated structure for spatially and temporally precise release, then what's the nature of the reception machinery that's able to respond to this fast dopamine release? And this is perplexing because dopamine receptors are G protein coupled, so they are slow compared to ionotropic receptors found at fast synapses. They basically belong to two broad families, D1 and D2 types. And this is what we are going to focus on because these are the most abundant types in the striatum. So a lot is known about the receptor biophysics, their downstream signaling, pharmacology, but their density and location relative to dopamine axons is still unclear. So what we know about receptor localization and activation comes from two broad categories of studies. One is immunoelectron microscopy, and the other are affinity-based models. Okay, so immuno-EM studies from over 2 decades ago and one more recently have shown that dopamine receptors are excluded from areas where axons make synaptic contacts with MSNs or medium spiny neurons, the main cell types of the striatum. And so it was found that their receptors are either found at perisynapses, which are defined as 100 nanometer zones outside of the synapse, or more so extrasynaptically. And thus there was no evidence provided for a synapse-like apposition of these receptors. The second evidence comes from affinity-based models, which, as the name suggests, are based on the low and high affinities of D1 and D2 type receptors, respectively, found from in vitro studies. So together with concentrations of dopamine from the site of release, which is fairly high, imaged to the site of release and diffuses as you move further away, possible distances of these receptors were calculated. And as a consequence, diagrams were constructed, leading to the view that D1 type receptors are closer to axons within 2 microns compared to D2Rs, which are further away and within 7 to 8 microns. However, recent experiments have shown that both types of dopamine receptors can exist in both low and high affinity states in vivo, suggesting a reconsideration of these affinity-based models that we discussed in our review, and overall urging the need for an experimental assessment of dopamine receptor arrangement in vivo. So this brings me to the overarching question for this project, which is, how is the dopamine receptor machinery organized? And for this, the first important question to address is, what is the spatial distribution and localization of the major dopamine receptors? So in order to visualize these receptors in the striatum, the methodology that I follow is shown here. I follow a typical immunostaining protocol where the animal is perfused, the brain is extracted and fixed in paraformaldehyde, and then 20 micron slices are made. I perform immunostaining with antibodies of interest on these slices and then image the dorsolateral striatum, again, because we're looking at the movement or the nigrostriatal pathway. So we obtained dopamine receptor antibodies published twice by this group, which was shown to work for basic confocal stainings, and I obtained them, knockout verified them to make sure they were specific before using them for sophisticated localization experiments. Here's a confocal staining, a basic staining for these two receptor antibodies. So I found that the signal from both antibodies was actually mutually exclusive, which is demonstrated here by a very low correlation coefficient, and also consistent from previous literature showing that these receptors are exclusively expressed on MSN. So either D1R would be expressed or D2R. So in confocal, we are right at the limit of resolution, which is around 200, 250 nanometers. And for this particular question, we ultimately want to analyze distances of these receptors relative to dopamine axons. So for this, we utilized 3D structured illumination microscopy, a super resolution imaging method with nearly double the lateral and axial resolution of confocal. And so I stain for D1 or D2 receptors for an antibody against the enzyme tyrosine hydroxylase, which is used for dopamine synthesis and hence labels dopamine axons. And on the extreme right is a merged image of either receptor with the dopamine axon, which is ultimately used for analysis. Here's the methodology that we use to analyze dopamine receptors relative to axons. So I acquire a 2-channel 3D image stack. And for the dopamine receptor channel, after some initial background subtraction and thresholding, I obtain all the voxels. And for the TH or the dopamine axon channel, we use segmentation or object recognition to get these axon objects. And then for each voxel of the dopamine receptor, we find what is the distance to the closest axon object. In order to plot this kind of distance distribution, where the y-axis shows the percentage of dopamine receptors within a particular distance from the axon object or from TH. And so the idea is that if a receptor type moves away from axons on average, then you would expect to see a rightward shift in the distribution for that receptor type. So the first thing I did was to compare these distance distributions for D1 versus D2 type of receptors relative to axons. And I found that D2Rs were actually closer to axons than D1Rs. So in green, I'm showing D1s, in magenta, D2. And here, looking at the distance distribution, you can see that D1R distribution is shifted towards the right with a peak of around 500 nanometers, whereas this is 300 nanometers for D2Rs. So this was a little surprising because the expectation from affinity-based models that I described before was that D1Rs are actually closer to dopamine axons, but what our results showed was that D2Rs were actually closer to dopamine axons than D1Rs. Okay, so next we wanted to understand what signal could possibly dictate this localization? Could it be release of the neurotransmitter dopamine itself? And so the experiment was to abolish dopamine release and then assess receptor localization. And for this, we wanted to use a condition where dopamine release was abolished. And so we used SIT1 mutants. So synaptotagmin 1 or SIT1 is the calcium sensor for fast presynaptic exocytosis. And a postdoc in our lab showed that knockout of SIT1, specifically in dopamine neurons, abolishes dopamine release as demonstrated here and measured using amperometry, shown here by a flat line compared to wild-type controls. So this condition provided a genetic tool to now abolish dopamine release and ask what happens to receptor localization. I did this, the staining protocol that I described and the analysis for D1 type receptors first. This is in littermate controls and knockouts for synaptotagmin 1. And actually, I found no significant change in the distance distributions in these two conditions for D1Rs. However, when I do this analysis for D2 type receptors, I found that D2Rs are actually further away on average from these axons in the absence of SIT1 or in the absence of release, suggesting that D2Rs sensed the absence of release and moved further away to change their localization. And this is shown here in magenta. So dopamine release itself could be a possible signal to tune dopamine receptor, specifically D2R localization. And we observed a differential response of these receptors to release where D1Rs were largely unaffected. So to conclude, I basically showed you that D2Rs are closer to axons compared to D1Rs, which is contrary to what's been previously expected out of these receptors. And absence of release, using synaptotagmin 1 knockouts in dopamine neurons moved D2Rs further away, specifically leaving D1Rs unaffected, showing that D2 type receptors are more sensitive to release than D1Rs. And so in the future, I would like to understand what is the mechanism that ultimately is controlling this localization. Could it be downstream dopamine receptor signaling? And we are trying to figure out tools to actually validate, to check this. And I would also like to do the inverse experiment of abolishing release, which is to enhance release in a particular part of the dopamine axon using a channel rhodopsin and stimulating to then assess whether receptors actually sense more dopamine and move closer to the site of more release. And finally, I also would like to knock out receptors specifically, so either knock out D2 type receptors or D1 or both to see how they affect presynaptic release in the system. So overall, just figuring out the precise arrangement of these receptors relative to release sites and axons would help to get a better understanding of this system comparable to what's known for glutamate and GABA, and also help design therapies which better match the timescales of this dopamine signaling. So finally, I'd like to thank all members of the Kaiser lab for providing a supportive environment for Pascal for all his mentorship, funding resources, Nikon Imaging Center for help with all the super resolution, and also Leading Edge for this wonderful opportunity. So thank you everyone for listening, and I'm happy to take any questions.
Thank you, Pragya. That was awesome. We do have some time for a couple questions. If anyone wants to raise your hand or speak up in the chat, I'll give you a sec. Okay, while we're waiting for people to think of some questions, I find this super fascinating. So I have lots of questions, but the first one I want to ask is, do you think that the dopamine receptor sort of localization and proximity could be affected by either previous experience or learning? And perhaps that's why people have seen different things. Maybe the animal's experience throughout their life changed, or even right before, you took the tissue might have changed this localization.
Yeah, I mean, that's an interesting point. And actually, definitely in terms of understanding and seeing precise arrangement, I think that's definitely a point to consider. As far as D1 versus D2 goes, and the localization and the distance plots that I showed goes, I think broadly speaking, we never see so much variability from animal to animal. The overall distributions and how they're located and microns are fairly comparable within different conditions, within different controls. Even between D1 versus D2. And even though it seems like there's just a 200 nanometer difference in the distance distributions of both, it's very conserved from, different conditions, different mutants. Sichuan is one condition that I showed you, but we've done this for other mutants as well. And so at least, even though the animals in the mouse colony are undergoing different kind of experiences, at least in that regard, we see fairly constant results. And the difference with previous studies is basically, so people haven't really assessed receptor localization as such. They've, extrapolated what might happen based on behavior or even on computational and mathematical modeling studies. So this, at least in our hands, using this super resolution method, seems to be pretty convincing and robust.
Yeah, absolutely. I suppose I'm wondering, like, if you could put the animals through an experience, let's say give them cocaine or something, because if it is due to increased dopamine release and they have gone through some sort of experiences, that would facilitate that. Yeah.
Definitely. Yeah, no, that's a very good point. And we're actually planning on doing some experiments with cocaine, also with levodopa, which is more relevant to the nitrostriatal pathway. But yeah, definitely great question. Following that up.
Okay, fantastic. Okay, any other questions? We'll give you a second. If not, I'll pass it back over to Deepa. Thank you, Pragya.
Thank you.
All right. Our next speaker is Adriane Otopalik from Rockefeller University, and they're going to be speaking to us about neural mechanisms of social behavior in Drosophila. All right, Adriane, whenever you're ready.
Can you see my slides okay? Can you hear me and see my slides? Yeah, okay, great. Hi, yeah, so I'm Adriane, and I'm broadly interested in the neurons and circuits that mediate social behavior in Drosophila or fruit flies. And one of the reasons why I find Drosophila to be an interesting venue to explore this is because they are remarkably social creatures, much like us as humans. So what I'm going to show you first is just a social interaction between a male and female Drosophila melanogaster flies. And what you may see here is that when the male encountered the female, he quickly sort of went through a behavioral switch and began utilizing a variety of behaviors to woo her. So he's extending a wing to produce a courtship song. He'll tap the female to taste her pheromones, and he'll sort of persistently chase her. And he'll do this, for many minutes, tens of minutes, until he successfully mounts and copulates with her. So during an episode of courtship, the male is presumably taking in multisensory cues from the female and translating these into a variety of behaviors to woo her. And although I'm interested in this sort of multiplexed set of sensory motor transformations, today I'm going to explore with you some of the circuits I've begun to describe that underlie the male's capacity to translate visual information from the female into locomotor pursuit, or his chase. So in that previous video, you saw that once courtship commenced, the male was able to keep a very tight distance to the female until copulation. So this is just a few millimeters away from her. And he is able to do this by coupling his forward and turning velocity to that of the female. So prior to courtship onset, the velocities of the two animals are relatively uncorrelated. But then once courtship commences, we see that they're tightly coupled. And one way to appreciate this quantitatively across courtship assays is to simply take the covariance in those two time series. And you can see that prior to courtship onset, when they are disengaged, there's a zero covariance between the two animals' velocities, but then once courtship begins, you see that this nice peak forms, and the male is able to react to the female motion with a lag of just 150 milliseconds. So the male follows the female's locomotion at a sub-second time scale. So this suggests that this is governed by a very robust visuomotor control loop in the male. He's presumably taking in information about the female's position in space, so her angular position and angular size in his field of view, and he's perhaps calculating some sort of error between her position at one time point and his goal position. So he wants to center her in his field of view. And by calculation of such an error, he might be then modulating his turning velocity and forward velocity. And this feeds back to his visual detection. And this sort of continues on loop until copulation commences. So as a postdoc, I've of the many questions I've begun to think about, two questions that I've sort of explored in my early years as a postdoc that I'm going to describe here are which are the visual pathways that are utilized in this computation by the male, and how is this visual information translated into appropriate motor output? So when I began my time in the Ruta lab, previous work had shown that there was a particular set of visual projection neurons, these LC10 neurons, that are small object detectors, and that when we silence these, the male is actually unable to track the female counterpart. So we see here that the male is pursuing the female, and when an LED turns on, these neurons are going to be silenced right now, and you'll see that the female, he's no longer able to track the female. So we can again quantify this effect by looking at the covariation in their velocities during control at baseline, as well as when these LC10 neurons are silenced, and we see that that peak is lost. And I, working along with two undergraduates from Hunter College, were able to do sort of a silencing screen and determine whether or not other LC populations or visual pathways are utilized during this locomotor pursuit. And so these data are summarized here. So first, I'll just point out that the covariations are high in the control situations, but then when LC10A is silenced, that locomotor coupling is lost. And then if we look at silencing of these many other LC populations, we see that while some of these lead to some sort of intermediate deficit in the male's ability to track the female, that LC10A really is sort of uniquely essential in this behavior. So with this knowledge in hand, I decided to map putative visuomotor circuits by looking at the neurons that are downstream of LC10. So I decided to pay particular attention to these descending neuron populations, which descend from the central brain into the ventral nerve cord, where motor sensors that control the different appendages of the animal reside. And so in order to do this, I took advantage of the female hemibrain, which is a publicly available connectome for those who work in Drosophila. And I found that, in fact, there are only about 9 or 10 descending neurons that lie downstream of LC10 visual neurons, and that these project to many different motor neural pil in the VNC. So of course, I was interested in whether or not these different descending neurons play different roles in the control of male locomotion or wing behaviors during courtship. And in order to probe this, I really wanted to be able to manipulate and record from these neurons during the ongoing act of courtship. And this is, of course, very difficult because it requires us to recapitulate that very complex social environment or interaction while recording from these underlying neurons. So in order to do this, I've taken advantage of a virtual reality system that was really first developed in the Yamamoto lab, but we have since really advanced this system in the Ruda lab with a lot of remarkable work by graduate student Tom Hinmarsh-Sten. And using this system, I'm able to situate the male fly on a spherical treadmill, and I present this male with a moving target that traverses his visual field much like a natural female. And what's really critical is that he's fixed in space, which means that I can access his the neurons and circuits underlying this behavior for calcium imaging or electrophysiology while he's engaged in the act of courting this fictive female target. So this is just an example of this system in action. You can see that the male is turning right, left, with the target, and he is extending his wings and vibrating to produce a courtship song. So from this data, or from this system, I can collect a very rich set of data, and they look something like this. So what I'm showing you here is that the target is turning on at this time point and from here on, and I've recorded here with GCAMP, or a calcium indicator, the activity of LC10 visual neurons, and you can see that they turn on when the target turns on. And not only that, they're on specifically when the male is turning left and right and tracking that target. And one measure that we use in the lab to quantify how well the male is pursuing that target is simply a tracking index, or a moving correlation between the male's heading and the position of that target. And of course, using software like Deep Lab Cut, I can look at when the male is extending his wings. So using this system, I've been able to record from this wide variety of descending neurons downstream of LC10A. And I'm just going to show you examples of what those look like. So when the target turns on, the male begins to track that target. We see LC10 is on, and then in different animals, we can appreciate that these many different descending neurons also turn on. But although all of these neurons are based on the connectome downstream of LC10, we see that not all of them are actually activated during this visually guided pursuit. So next, I want to know how these descending neurons may relate to the locomotor output of the male. And in order to do this, I need to move from a less periodic or regular stimulus to one that actually samples the full locomotor profile or locomotor set of strategies that the male can utilize during courtship pursuit. So I've taken advantage of a much more complex stimulus, and this allows me to better relate the activity in these different descending neurons with the steering behavior of the animal. And from these data, I can generate something like this. This is a filter for DNAO2 that relates the animal's steering behavior or heading with DNAO2 calcium activity. And basically what this is showing is that there is indeed a linear relationship between DNAO2's activity and the heading, such that the neuron's activity essentially precedes or predicts the steering behavior of the animal. And I suppose what's interesting about DNAO2, for example, is that although it is correlated with the steering behavior of the animal, even when the animal is not courting, the gain of this relationship is increased once courtship begins. So of course, I've generated these filters for all of these descending neurons, and they come in sort of different flavors. So DNAO1 shows sort of a similar relationship where it is linearly correlated with the steering behavior of the animal, and the gain is increased when courtship begins. However, DNAO2 is sort of unique in this regard, in that it's not really correlated with the steering behavior of the animal when the animal is just steering sort of freely in space. However, this neuron is specifically turned on and activated during courtship, sort of serving as a labeled line or a visuomotor circuit for the act of courtship pursuit. Okay, so what have I shown you? I've shown you that males tune their locomotion to their female counterparts during courtship, that multiple visual pathways perhaps contribute to male courtship pursuit, but that LC10A is sort of uniquely essential. I have outlined the many DNs that convey visual information from LC10 to the ventral nerve cord or downstream motor centers. However, not all of these DNs are recruited for courtship. And I've shown you that of those that are recruited, some are sort of steering generalists, whereas others are recruited in sort of a state-dependent manner, like that DNA10. And so taken together, I'm beginning to sort of flesh out the circuits underlying this sort of visuomotor control system for locomotor pursuit during courtship. And I think moving forward in my postdoc, I'm really interested in thinking about the many different intersecting control loops that mediate the many different behaviors that males perform during the ongoing act of courtship. And it will be interesting to see how the structure of these circuits may vary across these different component behaviors. And I guess before I close, I just wanted to mention that I've sort of presented you a very sterile situation, or a sterile instantiation of courtship. And I think it's really important to remember that Drosophila operate, in fact, in very complex social and sensory worlds. And I've sort of presented you this very simple task where a male pursues a female without any sort of competition. But of course, when a male is situated perhaps on a food substrate in these groups, males are, of course, attempting to fixate on their female of interest while also having to fend off and or avoid many other individuals in the same space. And so using these virtual reality systems, I think we're maybe perhaps capable of beginning to look at how these same circuits that I'm describing in this sort of simple situation, I'm now able to look at how these same circuits contend with these noisier social environments. In the case where then a male would have to sort of dynamically filter out other individuals with the hope of fixating and pursuing that target of interest. And of course, what makes these ethologically relevant tasks extremely complex is that these scenes are, of course, continuously changing in time. So moving into the future, it is my hope that Drosophila courtship could be, I could use it as a venue for the study of sort of this selective social attention and information flow. I think a set of sort of cognitive computations that have really been until recently kind of reserved for the cognitive capacities of much higher level primates. And it's interesting to think about how even the humble fruit fly is able to take on such complex tasks. Okay, and with that, I will just thank my mentor, Vanessa Ruda, and the many lab mates I have in the Ruda lab who have made it a really positive place to work. My funding, of course, from the Hannah Gray program, and I'll, of course, thank all of you for your attention, and we'll take any questions.
All right, thank you, Adriane. That was a wonderful talk. We have a question in the chat from Shweta. Do you have a sense as to how these DNs may vary between males and females?
That is an excellent question. We are still awaiting the male connectome, but I think it is going to be out soon, and that'll be a great help because one can imagine that these are, of course, sexually, like courtship is very sexually dimorphic sort of scenario to look at behavior. So one might expect that if they're going to be dimorphisms in the DNs that are recruited, or that you might expect to see them in this venue. We do know of at least a few male-specific descending neurons that are used during courtship. PIP10 is one of them. I didn't really talk about it today, and it's specific for courtship song or wing extensions in males. And there's a second one, ASP22, which is specific to males. I've imaged both of these on the virtual courtship setup, whereas PIP10 is recruited during this visually guided courtship. ASP22 does not appear to be active. So that's what I can tell you so far. But yeah, the sexual dimorphic aspect of this is definitely interesting to think about. Yeah.
Okay, and Julie has her hand raised, so go ahead, Julie.
Hey, thank you for this great talk. I really enjoy it. I had a quick question regarding like individual recognition in flies. Like when they're like tracking a female, you know, do they choose that female based on some orders, I guess, that she's receptive or what? And then after that, do they have like some visual cues of who is who, or like some other sensory cues? What is your understanding on that? How can they lock? Is that just locking on this and then that's the only cue, or do they have something else?
So this is something I'm really interested in pursuing on the virtual setup, kind of playing with activation of neurons that are thought to maybe kind of provide a representation of the male's arousal. But anyway, sorry, but specifically to answer your question, so there's a very rich literature looking at sort of pheromone detection and species-specific pheromones and how those guide male choice and identification of an appropriate mate. So I think in the scenario where there are multiple species present, which definitely happens in the wild, males will use these cues to determine who's appropriate. But I think what I kind of glossed over is, so even in like group scenarios where you have only one species present, where maybe you have multiple female targets, they're all virgins. Even then, so in that video that I didn't really point it out, I showed a video of group behavior, and you can actually follow pairs, male-female pairs in those group videos where a male actually does stay fixated on the same individual for tens of seconds, even though there are many other opportunities there for him to become distracted or begin interacting with other individuals. And so, yeah, I'm really interested in maybe taking advantage of the virtual system and activating some of the neurons that are known to become active when a male sort of initiates pursuit of the female, and to begin thinking about sort of whether or not that sort of visual information alone and his ability to track a female sort of reinforces his pursuit of certain individuals, and if the visual information is enough. Yeah.
Thanks.
Thank you. We should probably move on to the next speaker. Adrian, there is one more question in the chat, which you could probably answer, but yeah, we should probably move on to the next speaker.
Oh, can I? Yeah. Okay, thank you.
I think you could probably answer in the chat, Adrian, and that would be good. Okay, so moving on to our next speaker, we have Heather Snell from Einstein College of Medicine, and she's going to be talking to us about elucidating the mechanism underlying stress-induced motor dysfunction using a mouse model of episodic ataxia type 2. So take it away, Heather. Thank you so much.
So hopefully you can all see the slides and hear me. Thank you so much for staying and holding out and staying for my talk. And so I'm going to kind of jump into this so we can save some time. But our lab at Albert Einstein College of Medicine is interested in the cerebellum. And the cerebellum has been canonically known to be pivotal for coordinating and maintaining learned motor movement. It's this red part at the back of the brain here, the part that when most people are looking at the cortex in mice, they cut this part off and discard it, but we're really interested in understanding its function. Specifically, I'm interested in cerebellar Purkinje cells, which are these cells right here. They're the main output neuron of the cerebellar cortex. And what's really interesting about them, and also the cells that they synapse on, the deep cerebellar nuclei, is they are what's called intrinsically active, meaning that they can fire in the absence of synaptic input very regularly. And this is an example of a Purkinje cell firing here. They fire around 50 spikes per second. And it's this time invariant firing that allows them to encode motor-related information. When information comes in through one of these excitatory or inhibitory synapses, they're able to increase or decrease their firing based on these inputs. And that's how we can encode motor function at such a fast time scale. And this is something that was proposed over 60 years ago by John Eccles. And in this experiment, they put an electrode into the cerebellum and they task a monkey with modulating a lever with its wrist. And so this is the Purkinje cell firing here at rest. It's very regular, it's very regular. But once the monkey starts moving its wrist, the Purkinje cell starts modulating its activity based on this movement. And so this is true in health, but our lab is really interested in understanding what are the implications in this baseline firing, this very regular baseline firing in cerebellar disorders. And so we've looked at multiple cerebellar disorders. So I just kind of want to give you an example of how we looked at this. And so this is an example of just a normal ambulation of a person. And so we can start kind of breaking this down into some different variables. And so when we record from the cerebellum, either in vivo or in vitro, recording from Purkinje cells, similar to that monkey experiment, we can get our baseline firing at rest. And so this cell is firing around 65 spikes per second. And so our measure of irregularity is called the ISICV, or the interspike interval coefficient of variation. This is a standard deviation. So how different is each one of these spaces with the one before it and the one after it over the average over a period of the recording. And so at baseline, you can see that this is spiking at 65 spikes per second. The CV is low because the standard deviation is low. And so when the signal comes in, when the person starts walking, or when the mouse or the monkey start doing a task, then the signal comes in, and this downstream neuron from this Purkinje cell is able to discern between the baseline and the signal because they're easily discernible from each other. But when you have a model of ataxia, here is ataxia, uncoordinated movement, you have something that looks similar to this. And so at baseline, you have this highly irregular firing of Purkinje cells. And so this is, the average is similar to the wild type, but the standard deviation is going to be higher. So you have a baseline that has a wider distribution when you look at the firing rate than the baseline with the wild type. And so when a signal comes in, the downstream neuron isn't going to be able to as easily discern between what is the baseline and what is the signal. And that's one reason why we get this uncoordinated movement. And in fact, we've recorded from many different mouse models of dystonias and ataxias, which are cerebellar disorders. And we found that this ISICV can readily predict motor symptoms such as ataxias and dystonias, meaning a lower ISICV is normal ambulation and a higher ISICV is sustained motor dystonic postures. And so I'm really interested in understanding the mechanisms underlying the maintenance of this intrinsic activity, because I'm really interested in understanding models of ataxias and dystonias. And if we understand the mechanism of the maintenance, then we can start understanding what kind of therapeutic interventions can we give people with ataxias and dystonias to help their symptoms. So ataxia, again, being uncoordinated movement, and dystonia is actually co-contraction of agonist-antagonist muscles. I use a model called episodic ataxia type 2, which I'm going to tell you about in a minute. Additionally, in my independent work, I'm really interested in going further from this motor impairment, but also including the non-motor functions of the cerebellum to see how Purkinje cells and how cerebellar network can be implicated in non-motor functions such as intellectual disabilities using these mutations that lead to episodic ataxia type 2. And I'm going to tell you some about that too. And so episodic ataxia type 2 is a disorder in the CACNA1A gene. Here, every one of these rectangles is a mutation that results in episodic ataxia type 2. This gene encodes for the pore-forming subunit of a PQ-type Gaussian channel. This channel is expressed throughout the brain, but what's really interesting is it's only expressed postsynaptically in the cerebellum on Purkinje cells. Everywhere else is expressed presynaptically. And so this is really important for motor movement because we already know that Purkinje cell activity is really important for motor movement. And so patients with episodic ataxia type 2 experience these bouts of increased motor impairment when they're physically or emotionally stressed, when they consume caffeine, or when they consume alcohol. We have a model of this in a mouse called the tottering mouse, and they also experience this baseline tottering. That's why they got their name, this wide, incoordinated movement. They have a really wide gait, but they also experience these bouts of increased motor impairment as seen here with the twisting, this is dystonia, when you either physically stress them in a 50 ml conical tube, inject caffeine, or inject ethanol. And so we really wanted to understand what is the mechanism behind this increased motor impairment, so we can actually find therapeutics, because so far there's no therapeutics that can help treat these attacks. And so we got some clues from previous work in the lab, and we really wanted to know what channels are important. We know that this PQ type calcium channel is mutated, and it's a loss of function. So it's about a 40% decrease in the amount of calcium that comes in through this channel upon a voltage event. And we also know from previous work that this other channel, which is a calcium activated potassium channel, or SK channels, are both physically and chemically coupled to these voltage gated calcium channels. So calcium comes in, then that calcium activates these SK channels, which causes this regular firing of Purkinje cells. And so when we block this channel with a blocker called apamine, we can convert this cell from very regular firing to this highly irregular firing that we see. And so we hypothesized that these triggers, stress, caffeine, and alcohol, were actually blocking this SK channel, causing this increase in irregularity of firing of Purkinje cells and this increase in motor impairment. And so to begin to test this, we went into a head restrained in vivo recording model, electrophysiology. And so we implanted these head restraints on the mice and recorded extracellularly from the Purkinje cells and asked the question, what do these triggers, both caffeine and stress, do to the firing of Purkinje cells in this model? And so what we found was, when we measure spontaneous stress, which is just the stress from being on this head restraint for these mice, or when we injected caffeine, we saw an increase in the irregularity of firing of Purkinje cells to a similar extent. And this again is that ISICV, this measure of irregularity for both. And actually, this is at baseline, and this is just when you inject one of these. This is during spontaneous stress, but this looks very similar to what we saw when we blocked apamine in the previous slide that I showed you with the block of the SK channels. And so we hypothesized that indeed, these triggers are actually leading to this further block of SK channels. And we really wanted to understand a few questions after this. What is the mechanism behind these trigger-induced attacks, so this increase in motor impairment and this increase in Purkinje cell irregularity that we saw in vivo? And we had to start somewhere. We couldn't really attack all three of these at the same time. And you can, to a certain extent, abstain from alcohol, and these days life is stressful, so it's harder now. You can abstain from caffeine. You don't have to drink caffeine if you don't want to, but you can't really abstain from physical or emotional stress. And when you try to abstain from emotional stress, I feel like you get more stressed. At least I do. If I think about how stressed I am, I get more stressed. And so we really wanted to start with stress-induced attacks. And I'm going to go over some of this data. This paper just came out in Science Advances last month, so please go take a look at anything that I'm not really describing. And so to begin to understand this mechanism, we got some clues from previous work on other episodic channelopathies, because stress can release CRH, it can release norepinephrine. There's a bunch of different hormones that are involved when people are stressed. But we saw this work from Ellen Huss's group found that when you block the noradrenergic pathway, you can actually block attacks in episodic dysfunction. So we thought, okay, let's focus on norepinephrine. And then we got kind of lucky with this, because previous work in John Edelman's group found that just in normal environment, that norepinephrine can lead to the activation of alpha-1 adrenergic receptors, which actually leads to the reduction of SK channel activity. We're like, perfect. Maybe this is our mechanism that is happening in here, because they already have a reduced SK channel activity. They can't compensate, and it leads to this decrease in motor impairment. This actually happens through a kinase called CK2. This is casein kinase 2. This phosphorylates the calmodulin on SK channels, leading to a reduction in their function and impairment in their activity. And so we really wanted to go through each one of these mechanisms, each step in this mechanism, and kind of attack it and see if it's leading to these attacks. And so we started in vivo. And so our paradigm for restrained stress is popping the mouse in a 50 ml conical tube for 30 minutes. They get super stressed out, and then you take them out and you can assess their attacks. And so what we did is we did this, and we injected prazosin into the cerebellum of these mice. We found that this was sufficient to prevent these attacks that is caused by just restrained stress alone, suggesting that alpha-1 adrenergic receptors in the cerebellum are responsible for these attacks. Additionally, we wanted to make sure it was going specifically through the neuroadrenergic pathway in the cerebellum. And so we injected norepinephrine into the cerebellum and then injected prazosin, our alpha-1 adrenergic antagonist IP. And we found that that was also sufficient to prevent the attacks that norepinephrine caused. But I started this talk talking about in the ISICV and the firing of Purkinje cells, and you can't really do that in an in vivo preparation. So we switched, so we really wanted to understand, is this increasing the regularity of Purkinje cells of intrinsic pacemaking, so blocking synaptic input, and what are the receptors that underlie this? Is it going through these alpha-1 adrenergic receptors? And so we switched to an in vitro preparation, and we actually recorded extracellularly in Purkinje cells, and we blocked fast synaptic input from excitatory and inhibitory cells. And so we added picrotoxin, CGP, and CNQX, and we added norepinephrine to the bath and see what it did to our Purkinje cell activity. And what we found was norepinephrine caused a significant increase in the regularity of firing in both wild type and in Tautourine mice. And this is our ISICV, or measure of regularity. And when we added prazosin prior to adding norepinephrine, that prevented this increase in irregularity that we see. And so this suggests that indeed we're going through norepinephrine, we're going through alpha-1 adrenergic receptors, causing a decrease in SK channel activity and an increase in CK2 activity. But is this going through, and an increase in CK2 activity, but we haven't really proven that CK2 is involved. And so this is where I go, lead me to the therapeutic intervention. We were really lucky. We found this drug, CX-4945. It is FDA approved for treatment of cancer patients. And it's a potent and selective CK2 inhibitor. It's orally bioavailable, so we won't have to actually inject anything into the cerebellum of these people. They can just take a pill and it will work that way. And like I said, it's FDA approved, so we can fast track this for treatment of episodic ataxia type 2 in patients. And so what we did is we asked the question, can CX-4945 prevent this irregularity that we're seeing in Purkinje cell activity? And also, can oral administration of CX-4945 prevent stress-induced attacks? And indeed, that's what we found. When we added, similarly to our prazosin protocol, when we added CX-4945 prior to norepinephrine, it prevented that irregularity that we see with norepinephrine alone. And when we gavaged the mice with CX-4945 and put them in our stress paradigm, it prevented the attacks that we see with just stress alone. And so this is very awesome. We're very excited about this. I can tell you it also works for stress, or for caffeine and for ethanol. So we think this is something that we can fast track for patients. We're working on it right now to get this into clinical trials for patients that have episodic ataxia type 2. So just to summarize this, stress activates alpha-1 adrenergic receptors. It increases the activity of this kinase CK2, which further decreases SK channel activity, leading to aberrant Purkinje cell firing and the attacks that we're seeing. So with just like the next couple of minutes, something that I'm really interested in is a lot of these CAP1A mutations actually prevent with non-motor symptoms. So they have autism, they have intellectual disabilities, a lot of them have encephalopathies, and no one really understands the mechanism behind it. And what's also really interesting is the cerebellum has been highly implicated in autism, only second to having an autistic identical twin. And so I'm really interested in taking these CAP1A models and CAP1A mutations and really using them to understand what is the cerebellar contribution to autism, and specifically what is the CAP1A contribution to autism. So I submitted an R21 in collaboration with another PI in this department, Perry, who does C. elegans work, kind of taking 9 mutations that we know that haven't been characterized yet, and characterizing them that lead to intellectual disabilities, characterizing their expression and their function, and then also their expression, presynaptic expression in C. elegans. So that was submitted in a couple of weeks ago. And then additionally, I'm making a genetic mouse model. I have a genetic mouse model and a knock-in model of some of these mutations, and kind of doing a lot of behavior to look at their intellectual disabilities and to see if we can actually figure out ways to reverse the symptoms that we're seeing. And so with that, I'd like to thank the Leading Edge. I'd like to thank my collaborators and my amazing lab and all of the funders, Ford Foundation, SFN, and NINDS. So thank you.
Thank you so much, Heather. That was wonderful. And we can take any questions now that anyone might have in the chat, or go ahead and raise your hand. I think there's a lot there. Okay, let me give people a second. And while we're waiting for some questions, I do have a question. I'm sort of thinking about the level of stress, and I'm not really familiar with how the level of stress might contribute to the ataxia. But the general question is, does it need to be an acute, pretty intense stress, or have you looked at like lower level chronic stress or early life stress, which might also tie into your autism models?
Yeah, so with the patients of episodic ataxia type 2, normally it's acute stress. So it's like working out, or that's the physical stress is normally working out. And then the emotional stress is just having like an emotional event. So something will just happen, and then it leads them to having these attacks, which can actually last from a few hours to a few days. So it's mainly acute stress. No one has really looked at chronic stress to see if there's any kind of change in any kind of like norepinephrine pathway modulation or synaptic strength with acute stress. And that's something that I am really interested in looking at. But so far, all the work that we've done and all the work that the field has done has been like this very acute stress, like putting the animal into a 50 ml conical tube for a period of time. That's plenty. And when the animal model, you can just look at them wrong and they start having an attack. Like any kind of little bit of stress will cause them to have an attack. And that seems to be with patients. But there is also a huge phenotypic heterogeneity in patients. So some patients have attacks with just stress. Some people have attacks with just caffeine. Some people have attacks that last an hour. Some people have attacks that last a few days. So understanding this phenotypic heterogeneity is something that I really do want to tackle with my screenings, with the C. elegans and the heterologous expression system, to kind of understand the mechanism behind such a widespread of phenotypic heterogeneity in these disorders, so we can kind of start to get an understanding of what is the mechanism that might lead to each one of these symptoms.
Yeah, absolutely. I think maybe, the susceptibility to stress might play some role over time, and then potentially the ability of your treatment to have a successful outcome. So fantastic. And then Pragya has a question. We'll take one from you.
All right, thanks. Very nice talk, Heather. Very interesting. So I was wondering, I had a question about your initial stuff, regarding the cell firing, Purkinje cell firing. So what ultimately, I understand that there's mutation in the calcium channel, but is that the sole thing that's determining the change in the firing rate at baseline? Because you mentioned that those cells don't have any input and that's their intrinsic firing rate. So what are the driving factors that are changing that in these ataxias?
Yes, so at baseline, and I don't know if you noticed that, at baseline, the firing rate the regularity of these cells are regular. And we've done some previous work in the lab has shown that it's actually not the PQ-top calcium channel itself, but everything kind of leads back to this SK channel, this calcium activated potassium channel. And we've done work that's shown that just at baseline, if we activate the SK channel, then that can bring the ISICV down and improve some of the motor output of these mice just without any of these triggers. And so everything kind of leads back to this calcium activated potassium channel. And so we think that this is kind of a converging point, is the CK2 and SK channel modulation of all of these triggers, and of just the mechanism of the intrinsic activity of the firing of Purkinje cells, because we don't really know as a field what exactly is the mechanism that underlines how these cells can fire super regularly. Like, we know SK channels are important. We don't know the upstream mechanisms that lead to SK channel activation in just a normal wild type person or a normal human person. So that's something that I am actively working on. I have to do some more calcium imaging, but I'm almost done with that paper. So hopefully I can get that out soon. Kind of determining which receptors are important, what processes are important in the maintenance of this intrinsic activity of Purkinje cells. But everything converges on SK channels.
All right, wonderful, thank you.
Okay, it looks like we have one more question before we close out in the chat. says, amazing talk. Are these channels expressed outside of the nervous system, perhaps in cardiovascular or muscular tissues?
Yes, so they are expressed in the heart, and they are important in the maintenance of, so that's one of the really cool things is they're expressed in a lot of processes that have kind of intrinsic activity. And so you can think of like the heartbeat. It kind of beats on its own. It's kind of independent of anything else. And so it is really important in the heart. And I think it's also expressed in muscles, but mainly it's cardiovascular and it's neuronal. So nervous system and the cardiovascular. Yeah. And what's really good is the CK2, because it's already been FDA approved, it's been shown to have very little side effects. So one of the worries was, is there going to be cardiovascular side effects? Is there going to be other side effects? And they didn't see any. So hopefully this drug can be fast tracked and help these patients with very little side effects.
Fantastic. Thank you. Amazing work.
Thank you.
So with that, I would like to thank everyone so much for joining us today. And thank you to all of our wonderful Leading Edge fellows for your fantastic talks and contributing to the symposium. And if everyone would please join us for the next Leading Edge Fellows Systems Neuroscience session. It will be on July 18th at 12 p.m. Eastern Standard Time. Thank you, everybody.
Thanks, everyone.